Well, good afternoon. Thank you for joining us for the virtual 9th Annual Forensic Science Symposium Lunch and Learn poster session hosted by the Global Forensic and Justice Center. I want to let you know we are recording today's sessions and we'll make it available on GFJC's YouTube page. I will post a link in the chat for that, but it's go.fiu.edu/GFJC YouTube, all one word. For any questions you have, please use the chat feature. You can chat directly with myself, Danny Nichols, or with our presenter for any questions that you might have. Any questions that we aren't able to get to or that you may be missed because you're coming and going to posters will be provided in written format as quickly as we can. You can make this full screen presentation by clicking the 4 arrow icons in the top right-hand corner and clicking again to exit that full screen mode. With those housekeeping items out of the way, here's Mirna's presentation. It's all yours, Mirna.
Thank you so much, Danny, for the introduction. So my name is Mirna. I'm now starting my third year PhD in Dr. Bruce McCord's lab at FIU. And today I am presenting to you on the in-process development of qPCR TACMan assay to identify household miscellaneous biological mixtures. Before starting into what I did basically and what I got and what I will be doing in the future, I would briefly tell you why we need such type of an assay in a forensic application. Well, in many cases, a biological sample found in a crime scene may not in particularly be a human in origin. So what they've done, and when it's non-human, what they've done so far in casework is they would either run presumptive tests to tell you, oh, this blood, for example, is a human or non-human, but then this test would not particularly tell you what kind of species this non-human biofluid is. And other type of techniques, of course, started to be applied in forensic caseworks, but they're more labor intensive, they're expensive, or they take time. So we started with a technique called Pyro Sequencing, which is not the target, but I will briefly touch upon this one. So it's one of the types of next generation sequencing methods that I have some results that I will discuss with you in a minute here. But that technique basically is relatively cheap and super easy to use in a casework. You can run up to, for example, 48 samples in no more than 180 minutes. So the time it takes to give you the sequence is not long, and it is pretty sensitive. So I would go one step back here, and I'll tell you a little bit about the methodology. So we've collected different type of samples from at least 30, if not more, species that are more commonly found in a household. Let's say the pets that you have in a house, or any meat that you eat in a house, or any other critters that you can find around the house. So some of the pets, dogs and cats, horses, rabbits, parrots, and some meat that we eat, well, obviously chicken, pork, turkey, goat, and so on and so forth, and other types of fish as well. So we've collected different types of samples, and then we've extracted them using two main extraction methods. That one is automated by QIAGEN called EZ1, and that is pretty straightforward. It only takes 16 minutes for 6 samples to extract, and then you get a clean DNA sample. And then we quant them using an in-house vertebrate-specific primers and cyber green PCR kit by QIAGEN on a real-time machine, and then we get the quant, Then we amplify our target. And in this case, our target is on the mitochondria. And if you're familiar a little bit about the mitochondria, we decided to use this target. Well, it's not only that the literature has proved its uselessness in species identification, but also the number of mitochondria in a cell are much higher than just the nuclear DNA. So the probability of finding mitochondrial DNA in a forensic exhibit is much higher than finding nuclear DNA. And as we all know, those samples might not be pristine and we might not have enough. So by going into the mitochondria, we're having a higher chance of having a positive profile. So after we amplify this target, we would sequence it to have what we can see in this table over here. So a small target, which is a total of 104 bases. So it's a short fragment, and that can tell you the difference, or that can identify human versus other species that I've listed some of them here. And I'm sorry that it might be small, but if you're on your iPad, you can actually zoom, which is a nice feature. But basically what you have in this purplish box are the different meat that we can find in a household. In the blue part is the pets that like common pets that we can find in a household, and some type of fish. But these are just, I've cropped some of the sequences just because of the size of the poster, but it's 104 bases. And you can, just by looking at the colors, regardless what base is this, you can see the differences, like some insertions in some critters and some species, and some substitutions of different bases. And that when blasted or when compared to a reference database, it would tell you the predicted species that you have. Since this is a forensic application, then we needed also to follow the SWGDAM guidelines to see how sensitive this assay is. So, for example, how degraded a sample can be and still give you a positive result. So with this stability test, for example, we did take different types of DNA samples that we heated on a heat block at 95 degrees Celsius, which is pretty high temperature, for different times, so 10 minutes up to 25 minutes. And you can see in this gel picture the degradation level of the DNA, so it's pretty degraded. And still, when we sequenced those samples, we got completely positive, clear results. So that shows it's good to be implemented in a casework because of how stable it is to sequence those samples. For the sensitivity, we wanted to see how low of a quant we can go, and we went up to 59 nanograms. That's the highest that we went, but we will be trying to stretch it out and see if also going to amplify more than that. But for the lower limit, which is also very important in a forensic application, it goes as low as 23 picograms, which is very minute total input that is required to get a sequence. So we got, and this is in the process of being published, we got pretty interesting results on different types of species. I have to say though, since this is one target only, it wouldn't distinguish a subspecies. So it wouldn't differentiate, for example, a Chihuahua dog versus a bulldog. It would tell me, yeah, this is a dog, but not necessarily what breed it is, for example, which is, as a forensic application, as a first step, it would be more critical as an investigative lead to tell me if it's a dog versus a human. There are caseworks that I was reading the other day, for example, that they found some meat or some skin in a fridge or a freezer, and they thought this is a human skin. When they actually analyzed it, it turned out to be a pork skin, for example. but why we have moved now to apply or to try the qPCR TaqMan assay. So the Pyro sequencing works the best for single sources. It does work for some mixtures, but it requires some manual evaluation. So we thought that maybe we can add another feature, which basically is a TaqMan assay, which is a real-time PCR assay that you design specific probes as you see here for your targets. And then if you have a mixture, let's say of a human and a cat, and then you can see those two, both of those two amplify, and you can tell that yes, this is a human and a cat mixture, for example. So different criteria we're following, stringent criteria we're following for multiplexing and adding different probes together. That is the melting temperature, the GC content, as well as how many Gs and Cs we have at the 3' end, so we don't create a hairpin, what's the amplicon size, and like so many different features. And then most importantly, we need to check any potential autodimer formation, which is basically when the primers and the probes bind together and not amplify your target. We don't want that, or any hairpin formation, because that will affect the efficiency of this assay. And then we would be running them multiplex, and I'm saying we would be because of the COVID, we've a little bit we're a little bit behind in finishing up this part, but we first test the single probes individually and then multiplex them together to optimize it at the end. So I don't want to take more time. I want to leave definitely time for questions. Thank you for attending this poster. And here, I would want to, at the end, thank QIAGEN for supporting the first part of this research, and the International Forensic Research Institute at FIU to be funding the second part of this research. With no further ado, the last thing that I want to say, this is my LinkedIn. So if you have your phone, you can go to your camera, and then it will directly give you a link to my LinkedIn. And here you can find a YouTube video that I've presented more details about the first part of this research that you can find in case you're interested in knowing more information.
Thank you, Mirna. That's really interesting. If anybody has any questions or would like clarification on anything, please feel free to put it in the chat and direct it either to myself or directly to Mirna, and we can get those questions answered for you. Mirna, I have one question for you. And why did you decide to use the TaqMan assay in addition to Pyro sequencing?
That's a very good question. So as I said, like I touched a little bit about it when I was explaining. So the Pyro sequencing is a very robust, easy, and relatively cheaper method to use, not cheaper than TaqMan, but compared to any other next-gen sequencing chemistries, to sequence small targets up to 125 bases. And it was giving us beautiful results, and we did try it on mixtures. It requires a little bit of manual work, but when the mixture get complicated, like a ratio of, let's say, 100 versus 1 or whatever other types of ratios, it would be challenging to decipher the minor contributor, for example. So we thought by adding this step, which is based on designing specific probes for our targets, then we can use it as alternative method to quickly resolve mixtures for human versus non-human, in case we find weird amplification in this step. Then we can do this one to give us a quick idea about what is the mixture composed of.
Okay, great. We'll give the guests a few minutes to read over the poster and let us know if you guys have any questions. Mirna, we have a couple of questions that have just come in. First, can you explain your approach to primer design and TM?
Yes. So when we design a TACMAN assay, we're designing two things. One is the forward and the reverse primers, and the second thing is the probe. For the forward and reverse, they're just regular primers up to up to so it has, like, the research showed that the primers usually are around 20 bases in length, and when multiplexing you can multiplex up to 8 primers at a time and 4 probes together. So that would give you 4 targets, because for each target, if you're taking a different region, let's say in the mitochondria, then you can design for each species, in this case, their specific forward and reverse, but most importantly, which is longer in length, the size of it. So it's between, the probe usually is between 22 and 30 nucleotides. And when we look at the melting temperature, the probe has to be 10 degrees higher than the primers to have sensitive and efficient amplification. So we try, when we design those probes and primers, to put these features as requirement for the design. So it would be it is challenging, in fact, to find targets that have so many guidelines. One is the size of the probe. Second is the melting temperature of the probe and primers, how many G's and C's you have in those primers and probes, as well as the total amplicon size. So it's all the way between the forward and the reverse, including the probe and the flanking region. So once we design those, we have to check if there would be any potential, using some softwares that are available online, if there is any potential for primers to be binding to each other, or the primers and probes to be binding to each other, because that will decrease the efficiency of the assay. And we don't want to have any autotimer or any hairpin formation. And the hairpin is basically when the two ends of a primer bind together and form a shape of a loop. So we try to avoid that too. So that goes into the designing. Another additional thing that we have to keep in mind is how many G's and C's we have at the 3' end of our sequence, because there shouldn't be more than 2 as a max, and at least the first five nucleotides. So once we have those features in, and we check for specificity, because we don't want that probe to be amplifying any target, right? We want that probe to be specific to our species of interest. And then at that stage, once you design them and you purchase them, then you start to test different designs to come up with the best design possible for this assay.
Thank you. One other question came in, and it's, what difficulties did you face on the optimization process?
So I would talk more about the Pyro Sequencing, because that's where I did the actual lab work so far. In the Pyro Sequencing, so all the way through, when I reached the quantitation part, I did find kits, ready-made kits that can quant human specifically. There are kits that can quant DNA in general, but that can include bacterial DNA, like a bunch of different DNA together, and that it gives you the quant. However, I didn't find any readily available kit, to the best of my knowledge, that can quant just vertebrate specific, because I don't want to quant bacterial and it tell me, oh, you have 100 nanogram, for example, of DNA, and it ends up being just bacterial DNA. And then I would be wasting my reagents when I do Pyro Sequencing. So we did a little bit of back and forth in that quant, where we designed primers that are just specific for vertebrates, and then quanting those for the amplification and the sequencing part. In the sequencing, we did design different types of primers to come up with the best potential primers that can basically sequence the DNA of interest with no preference over one mixture over the other. So that's more into the mixtures part, and that's why I said can they convolute some sort of mixtures. So some primers might be preferentially amplifying human over dog, for example, but we want to get the one that can amplify both in the same level and not preferentially amplify one another. So we were able to tweak those primers to give us the results that we wanted.
Great, thanks. If anyone was interested in those links and either couldn't or hasn't had a chance yet to get those QR codes to work, I did paste each link in the chat for you to be able to copy and paste from if you want to check those out later.
Thank you, Dan.
If anybody has joined us in the last few minutes, you have any questions, please feel free to put those in the chat, either to myself or Mirna, and we can get to those and be happy to answer them.
Maybe while we're waiting for some questions to come in, I just noticed that I didn't talk about why would we need a positive control in our mixes in attackman assay. So when we design attackman assay, we have to make sure that our reagents, for example, are working just fine, but let's say if we got negative results, it's just because we don't have any of our species of interest. So let's say I have a mixture that I know it's some bunch of different species together, 2 or whatever, and then I run this assay, but then I got negative. How would I make sure that the negative is just because I don't have any human, any cow, any cat or dog or chicken, but rather it's the reagents that are not working, for example. So in this case, we have to have what we call a positive control, which we know for a fact it should be amplifying every time we run this assay. So that way we can control our reagents and to see, to make sure that our reagents are still working fine and that it's just like we don't have any of those species of interest in our sample. The other thing that I would like to add, you're seeing here different colors, and that's on purpose, because when you're multiplexing, you have to make sure that each probe of interest has a different fluorophore. Or I should have added a video, but if we quickly search for TACMAN, it will explain why we have let's say a fluorophore and a quencher. So how it works, if we look back here, so let's say my sample has human DNA in it, and I added this the primer specific for human and the probe that is specific for human. If I have a human DNA, then my probe will come and bind on the target. Once it binds, that will lead to let the quencher out because it binds, then the fluorophore will emit some light, and that light will be measured in a real-time PCR like the rotor gene here. So in case I don't have human DNA, but I have the probe, it wouldn't bind. That means there wouldn't be any amplification. So the quencher will hide the light that can be emitted from my fluorophore. So I wouldn't see light emitted, and that's when I know that I have a negative result.
Great, thanks. We do have another question that just came in, and they are asking, what type of advice would you give to researchers when they want to design a sequence or probe?
That's a good question. Well, first of all, you have to make sure that you are using a curated reference sample, because all of the hard work that you will put in designing will be not good if you are working with a reference that is not good quality. So if there are any mismatches, or if there are any insertions or deletions that are not really there in a sample, and you're designing based off of that, then you will not get specific primers and probes. So one is make sure that you have a curated database or a reference sample that you are using as a reference to design your primers and probes. Number two, especially for probes, it's always suggested that to avoid any locations in the genome that has what we call variations between within the species of interest. So let's say I'm designing a cat probe. I have to make sure that where I am designing that probe, there are no polymorphisms or differences on that specific target between different cats, because it might work for one cat and not the other, and this will not be making my assay efficient. But it's good to have differences between those species in this context. So if I have differences between human and cat, and I'm designing it just based off of the cat, then it would be specific for my cat. So that's another thing to make sure that your probe is specific for the species of interest. When it comes to designing a specifically attackman in specific, not generally any assay, as I said previously, the primer size is usually between 15 to 25, ideally 20 nucleotides in length. And the probe size is usually between 22 and 30 nucleotides and ideally 25 bases. But most importantly is the melting temperature. That has to be around 10 degrees more for the probe than the primer to make sure you have an efficient amplification. So depending on what type of assay you're running, is it just normal a cyber-green assay, qPCR assay, or is it for sequencing, or is it for attackman? Then there would be additional requirements for each one that would be different, of course.
Any other questions? Feel free to put them in the chat. We're about halfway through, Mirna. Maybe spend 4 or 5 minutes in just going over the basics of your poster again, in case we have any new folks or folks that might just want a little bit of intro to it for us.
Okay, I'm gonna try to use different words, so in case if anyone is still here and they don't want to hear the same thing. So, do I start by my name? No, you already know my name. So this project is target is basically to identify non-human miscellaneous samples that you can find in a crime scene. As we all know, well, maybe now I'm using the same words, I don't know. And as we all know, in a crime scene, a biological sample might not be just human. It can be mixture, it can be your pet that you have in the house, it can be the meat that you eat. So it's important when we have such sample to know what species it is, to give you an investigative lead. Well, I just remember the case, I forgot where someone has told me, they're like, oh, you know that the O.J. Simpson case back in the 1992 or 93, where I was just born, I think. They had a mixture of human and non-human, and at the time there was no readily available validated kit in the market to know what was the non-human DNA. And it would have been nice if we were important to tell me, okay, yeah, they had a dog, and that blood that was found in this crime scene is the dog DNA, basically. So it is important to have an easy and quick method to identify those non-human DNA. So we started looking at the literature to see what targets can we use, how we can approach it, and it was we found consensus over using mitochondrial versus nuclear DNA. So mitochondrial DNA is found in much higher abundance, so that's good if we have a minute sample in a crime scene, and we will have a positive result if we are looking at the mitochondrial. What's cool about this first part of the the project is when we designed those primers, they're universal for all of our vertebrates. So we didn't need to design multiple or multiplex anything for the pyro sequencing. It's a universal forward and reverse primers that can be used to amplify around 104 bases for different species. And then by comparing them to a reference database, it can tell me a human versus non-human. And in particular, I have in this area here, the meats that we can eat: chicken, pig, turkey, goat, cow, and then some pets that we might have in a house: dog, cat, or a horse or a rabbit, and some fish as well. And you, just by looking at the color, you can see there are differences already between those species. We looked more into depth to see how stable it is. So we heated the DNA up to 95 degrees and up to 25 minutes, and we can see how degraded those samples are. And we found that even while degraded, it can give us also positive results. And for the sensitivity, it goes as low as 23 picograms to give us full profile as well. But we did try also some mixtures, and we did tweak our primers to make sure that they don't do any preferential amplification for one species over the other. And we were able to deconvolute mixtures up to some extent by using some manual Excel sheets that we created to come up with a conclusion. However, it might be troublesome to deconvolute complicated mixtures, and that's why we decided to go with a quick qPCR TaqMan assay that can be amplifying some targets of interest that are specific for our species. And in this case, it's a human, cow, cat, dog, and chicken, and a positive control to come up with an alternative quick method to resolve mixtures of human and non-human biofluids found in a crime scene.
Any questions? Feel free to put them in the chat. I have one here for you. That's, what type of samples did you test?
Yeah, we went all the way to the all extremes or possible samples that we can find. So we started with buccal swabs of living animals. I would say that was interesting because I went to, before all of the COVID-19 issues and all of the pandemic that we're now in, I went to a cattle and horse show in Miami, and there were a bunch of different animals. And then I started asking, so I do, of course, have the approval from the animal care facility at FAU. So I was asking owners, can I swab, buccal swab, like from the mouth, some saliva of those animals, like some horses, beautiful horses, and so on. So yeah, we have buccal swabs, and then we have also livestock meat. So we went to supermarkets, and then we purchased some meat, different type of meat from the store. That is important to bring another alternative application, which is the food market, but I'm not going through that now. But you can tell a pork versus a beef meat, basically. And so that was the second type of sample that we've extracted DNA from. We did have some blood sample back from the 2000s from the zoo of chimpanzees, monkeys, squirrels, lemurs, and so on, that also gave us a food profile. Then we went to test DNA from hair samples. So there is a myth on, oh, we wouldn't find enough DNA in a hair, in a rootless hair, so you don't even have the root. Well, in fact, that's a myth. There is a blog that I wrote the other day that you can find on my LinkedIn. There is DNA, it's just degraded, but for our target, because it's small, it gives you a full profile as well. So that's hair. We did try also teeth, DNA from teeth, and DNA from bone. And I have to shout out a thank you for Dr. Sato for helping me out in the teeth and the bone DNA extraction. That was also interesting to do. So yeah, I think all kind of biological evidence that you probably can find in a crime case, we've already tried this assay on. Just to break some silence in here, I would love to answer any question So don't feel intimidated to send any question, or if not, we can keep in touch to tell you the progress in this part on my LinkedIn. That Danny has already shared the link on the chat. If not, you can just take your phone, go to the camera without clicking anything. It would recognize the QR code. It takes you, gives you the link to go directly to the LinkedIn.
That's perfect. I was about to say the exact same thing.
Glad to hear.
We do have, I think you already touched on this, but one question, how can your research, this work be used in casework?
Yes, I think it's in the heart of caseworks. No, maybe I'm biased, no. So how can it be used in a casework? Well, in any case that you have, you know that you have a non-human DNA, because let's say you ran the quant for human DNA, and let's say you didn't find enough, or let's say you know that it's a mixture just by some presumptive tests that you ran, some antigen, antibody test, whatever, then you know that it's non-human. You want to know what species because it might help you in giving an investigative lead in your casework. Then by running this relatively easy and straightforward assay, in particular the Pyro sequencing, if it's a single source, then you would get the results in like 2 hours. Well, a little bit more if you're doing extraction, quant, and amp, but within a day or so, you would get the result of what is the, who contributed that non-human DNA sample, basically. And for the TACMan, in case you have a mixture, it will give you a better idea of what is the composition of your mixture, and also relatively very easy and quick way of running the assay. So I actually, when I presented this work in the ISHI and the AAFS conference, I had a couple of people who work in crime labs who approached me and asked if this assay is already in the market, or if there is any way for them to send us samples for us to test them and to tell them what is the composition of the DNA sample that they have. So we did see direct impact in the, I can say, in the forensic community. I would take the advantage now to say that we are willing to have any samples that you might have and you want to test to validate in our assay. This part is in the process of being written to hopefully being published soon, and this one will be, once we go back to labs, will be also finalized, hopefully. So if you have samples, please feel free to reach out for us. I should have added the link for the website for Dr. Bruce McCord. But if you reach out to me on LinkedIn, then I can put you in contact with Dr. McCord, who's a great support, and he loves collaborating. So we can go from there.
Great. Well, I will take this time to say that we have a little more than 15 minutes left. So if you do have any questions, now's the time. Get those into the chat, and we'll go over those. And then these will be, any of these questions, whether even if we run out of time or there were some asked before you got here, they'll be made available written format after the poster presentation. And we will also be posting the recording of this meeting on GFJC's YouTube page later on, so keep an eye out for that. If you're curious where that is, there's a link in the chat that you can access that YouTube page. So get those questions in. Mirna, I have a kind of a personal question on the poster. Were there any animals that you were hoping to do or wanted to do that you didn't get a chance to?
That's a good question. Let me remember. Speaking of which, I will try to answer this question, but if anyone has any type of sample that they're willing to give us an aliquot with, please feel free to send me a message, and then we can we can chat more about it. But the type of samples, let's see. Well, I did have rats, but I was trying to get some mice, which should be easy. Like we go just to any lab that does mice DNA, but up till now, because of the lockdown, I had difficulties in finding mice DNA. But I did try a couple of different fish, so many dogs and cats, horses as well. I even have chimpanzees and monkeys DNA. So I think other than the mice, I don't think there would be, or maybe something that I didn't think about it. But yeah, if you have any sample, anyone has any pets or any samples that are willing to share an aliquot with, it doesn't have to be extracted, it can be the actual tissue or anything, then we can communicate to collaborate on that.
Do you see any maybe any future needs for maybe seeing certain subspecies or anything like that for this?
Well, that's a very important question. Depending on what is the aim. So is it the aim of, for example, in pouching or in wildlife forensics? It would be for them important to know what subspecies you have. Let's say you're talking about the fish market and you want to know if it's a breed that is not allowed to be poached or fished, then you want to know the exact subspecies. In that case, once you know that it's a fish DNA, for example, then you go further into different types of assays that you can find that would tell you the subspecies. But before going all the way to the subspecies assays that you're using, you might need to do this step first to know if you actually have a fish or whatever other biological DNA sample you have. So yes, it would be important in some disciplines and in some aims, but that would be an additional test that you can do. And I'm pretty sure there are some companies, for example, for dog breeds, that can tell you a breed over the other, in case you're interested in that. But there is constant research on barcoding for the species present that can be used for an additional assay.
Great, thanks. If anybody, we'll give you guys a few minutes to read over the poster. If anybody has any questions, throw them in the chat.
Just to break here the silence a little bit, I thought I would say a non-science fact in this poster. So in case you're wondering where these icons I got from, well, PowerPoint, luckily they've updated their program, and now easily you can find the icons, and then you just type in what you want, and they have a good variety of things, but I found my animals, so I was happy.
If anybody's joined us in the last few minutes and maybe missed the basic intro to the poster, feel free to let us know. Mirna can go back over that or ask any specific questions that you have, just say so in the chat for us.
So I have a question here for repeating the intro for people who just joined. And briefly, this is the study of developing a Tachman assay to identify non-human miscellaneous mixtures. And we started using, first of all, a different technique, which is Pyro Sequencing, that is relatively easy and quick method to amplify targets. And in this case, it's 104 bases. I just cropped it for the sake of the space in this poster. But you can see that just by amplifying one target with universal forward and reverse primers, you can see the differences between different species. And we're moving on to designing a TACMAN assay, multiplexing different species of interest, starting with those 5 species as a startup, because they're commonly found in a household, to deconvolute mixtures that might be challenging using the first technique, which is the Pyro Sequencing. So this part here is in the process of being published, and this part is we're finishing designing now, and we're starting soon with the experiments for optimizing this assay.
I may have missed it earlier. You may have said it, but about how many samples did you process?
Oh, that's a good question. Well, I had at least, if not more, 30 species, but I had more than 1 sample per many of those species. So at least I can say I've processed around 180 different samples from around 30 species.
Wow, that's great.
Mm-hmm. And for the primers, for the Pyro Sequencing primers, it was a collaborative work between Frank from QIAGEN and myself. And it was a great teamwork that we worked on for the Pyro Sequencing assay design. For the TACMAN, it's more, I'm looking more into literature and into designing my own primers and my own probes. Yes, so it is in-house designing. And from the literature, it says that we can go up to 8 primers and 4 probes in a master mix, in one master mix, using 4 different fluorophores and basically 4 pairs of primers. That is a total of 8 primers in a master mix. We're starting only with 3 and not 4 per master mix, just as a startup, but that would be able to be adding 1 more probe in each master mix, for example. But for our budget limitation, we're starting with those 6 targets. As I said, for the positive control, it's important just to make sure that our reagents just work just fine. If we get any negatives in the others, that means we don't have any of the others, and not that the reagents are not working.
Well, great. Well, we have about 5 minutes before the end of the poster session. So I just want to ask everybody to get their last minute questions in. If there's anything else that you would like to ask Mirna, now is the time. Otherwise, once you're done reading your poster, be sure to return back to the main webinar where you'll get some more instruction this afternoon.
I really appreciate each and every person who joined, who are joining still and joined already this poster. It means a lot to me to stay connected. So on the left upper corner, you can find the QR code for my LinkedIn. And on the right-hand side, you can find a QR code for a YouTube video recorded by ISHI conference when I was presenting the first part of this research over there, so you can get more details in case you've missed anything. And Danny has already shared the links for those videos, for those 2 QR codes in case you've missed them. And I think that's it.
All right, Mirna, we have one final question here. It is, what software did you use for sequence alignment?
For sequencing alignment, yes, there is software called Muscle, like our muscles, MUSCLE. If you're Googling it, if you just type in muscle, you're gonna see a lot of muscles pictures. So instead of that, just type in muscle alignment software, and it will take you to the software. It's super easy online service that and relatively very quick. You only need an input of sequencing data that basically a FASTA format or any kind of format, but the naming starts with well, they have an input template that you can use, but then you have the name, and then underneath it on another line you have the sequence, and then after that another sequence and the name, and then the actual sequence, and then it would do the alignment for you. It has different options of what type of output file you need, you can have, either colored or non-colored. Usually I take it non-colored because I can create my own colored scheme on Excel, because usually it's just weird looking colors. But generally speaking, this software is, or this online tool is super easy to use. So again, the name of the tool is called Muscle, MUSCLE. Just type in Google muscle alignment software and it will pop up.
All right, great, thank you. I think that will end our poster session today. We appreciate everybody for joining us. Mirna, thank you for your time. And everybody have a great day.